Thank you, far too kind. I didn't think we would make it. 10 episodes. It's Ralph from the South. It's the host that does the most, and I don't mean to boast, but I do because I'm the most excited because I have my best friends. On today's episode, I have for you a wonderful gift. It's a Buchmueller lab reunion featuring my two best friends, lab mates, Haley Stubbs, Daniela Mesa Sanchez. We're all fifth-year grad students. We're all in different places, with Haley being at Vanderbilt University in the Quantitative and Chemical Biology program, and my friend Daniela Mesa Sanchez in the Department of Chemistry. It's amazing to have these two geniuses on with me. We got double the guests, double the time, double the rhymes. Well, maybe not so much that last part, but it's all right. Today's episode: natural highs and lows of scientific research on Scientifically Sound. Let's go! And I'll take to the sky on a natural high.
Maybe you just smell too much chloroform.
Jesus! Oh, I die! Take to the sky on loving you. You know what? Oh, God.
You didn't even get to the next line. I had one ready for you.
You might as well say it now.
Oh my God, who is this?
Yeah.
Oh my gosh, who is singing so funny?
Yeah. What we got? We got science. We got music. Put them together, let's use it. Wait. Let me be the substrate in your ear receptors, and let this not just be one endeavor. I get crunk on the science, I pull the lever. And with the music, I just hope you say, dang, you clever. Whatever, bring it, master what matters. All the while my life is scattered. You surge on in general, I guess you're David Satcher. And you bet I got you when it comes down to the laughter and whatever may come after. Scientifically sound so profound, gonna convert energy like it's dollar to the pound. Just listen to this while you're making some hash browns, or maybe later after you raking some cash mounds. Who am I? It's Ralph from the South, who raps out his mouth, and we bring the music to the science that's around. And that's on period. And I just thank you for hearing this Scientifically Sound.
Oh my God, I'm so excited. I'm so excited.
Hello, everyone! What's up? I don't do it really well, so edit, undo. Whoever you may be, you know who it is. It's Ralph from the South. I am now so excited because I have my two lovely bays, my sisters, these wonderful women of STEM on this episode. To give some background of how we met, it was in a lovely time in Lakeside at Furman University. They're from Judson Hall, I, a lonely boy, in Towns Hall. And I would get to know them individually, but we really didn't hit it off until we entered this boot camp type class called Techniques, where we had to learn how to do synthesis for chemistry, analyze stuff for chemistry, cry a little bit, get back to analyzing once again. And these two, comically, would drop pipettes into columns that were used to separate out solutions. Not once, not twice, probably 3 times or 5 times. I don't know. It doesn't matter. But when it really hit it off was when we got together and formed the super team that we were. When we joined our lovely advisor, and if she's listening, Karen Buchmueller, we love you. We would always hang around in the office space, whether it was watching Great British Bake Off or saying yes to the dress. And I don't know anything about those things. They blast Disney music while they're doing gels. They'll roast me about anything when it comes to relationships. These two lovely people I can call dearly my best friends. Or better than that, we're more than friends. We're lab mates. ladies, everyone, ladies, gentlemen, non-binary people, give it up for Haley Stubbs and Daniela Mesa Sanchez. Woo!
Hello.
Wow, what an intro. What an intro.
I thought y'all would like it. How do we
So
feel?
many memories.
Oh, were they good or bad?
They were good.
Oh, great. Yeah.
They gave me a smile.
I'm glad, because all I could remember more, but we don't have to get into that until we really get into the nitty-gritty of things today. So I can only introduce you in one way, but you can truly introduce yourself on your own terms. So I'd let y'all introduce yourselves. Tell us who you are, where you're from, where you're at right now, and where did you grow up?
All right. So I'm Daniela Mesa Sanchez. I'll do it with the accent. I am originally from Colombia, but I mostly grew up in South Carolina in Greenville, where we all went to school. And I am currently in the middle of nowhere, Indiana, aka home of Purdue University, West Lafayette. And yeah, that's where I'm at.
Go ahead, Haley.
My name's Haley, and I am currently at Vanderbilt University in Nashville, Tennessee, good old Nash Vegas. Bachelor and bachelorette party capital of the world here. And I grew up in Roswell, Georgia, which is actually pretty close to where Ralph grew up. So it's interesting that we didn't meet each other until college. But basically in the burbs of Atlanta.
Y'all hear my beautiful bays be geniuses, one at Purdue, one at Vanderbilt. I love them. And they smart. They smart out here. So tell me, what made you get into science? Where did the love fall for you? I've heard a number of things, whether it's high school shenanigans or maybe when you were 5, you tripped and saw a microscope for the very first time. So since Daniela started us off, Haley, you start us off for this question.
Okay. I think initially I just ended up being good at chemistry in high school. I liked that there was a little bit of math involved. I just liked it. There wasn't really a solid reason behind it, which isn't very exciting. But and then I think once I got to college, I kind of fell in love a little bit with protein biology and specifically GPCR signaling. So I actually have a retinal degeneration, and it kind of created a fascination for rhodopsin, which is kind of what I like to think of as the OG GPCR. And so then I wanted to pursue GPCR signaling in my graduate career. And I kind of decided to go down the route of structural biology. I think structures are super fascinating. They answer so many questions. My advisor always says filling in electron density is kind of like playing a video game. So it's fun. It's really, I think, visually engaging. So that's kind of where I ended up today.
So all she needed was some structure, everybody. Also, for those who don't know, GPCR is G protein coupled receptors. Go look them up. They're vital to our living. All right, so you fell in love because you were good at it. What about you, Table?
So I don't know. I think I'm a little bit like Haley. I mean, I guess as a kid, I had this one microscope set, and I just made test tubes of dirt and ran around thinking I was going to be a mad scientist like Dexter. The cartoon, not the serial killer, just to be clear.
Allegedly.
And and then, yeah, in high school, I had this crazy chemistry teacher. He was like a legend in school, Mr. Dalby, Dr. Dalby, technically. He had his PhD, and, you know, there was like all this lore about him. He had like this weird... he like used to be a trade ambassador in South Africa and owned like a drill bit company that mined diamonds or some nonsense. I don't know. he was just like this mysterious figure, and he was super harsh in chemistry, and it was just like the most interesting class because everything else in high school was kind of like, okay, low effort, boring, but this guy was like a whole mystery, and he was super harsh. And so I was like, okay, this is cool. Like, this is interesting. And I guess at Furman, I was like, okay, I guess being a chemist sounds cool. And I think I was in the middle of it by the time that I even really questioned. Is like, is this what I want to do? And I was like, yeah, no, this is fun. We'll stick with this. And, you know, I'd already met our advisor by then, Karen. And I've kind of fallen in love with being in the lab and trying to figure out what went wrong and trying to dissect why things were not working. And whenever we were reading papers in lab, like, I think the thing that captured me the most were like assays and techniques and like the cleverness of how we use the knowledge that we have to figure out things that we don't know. And so that's kind of why I ended up going the analytical chemistry route and thinking about all these different instruments and techniques that we're able to use. And I don't know, I'm just always very captured by the cleverness of it. And I guess within that realm, there is mass spectrometry, which to me seems like this huge technique that is the most applicable out of everything. I mean, not bashing on all of the other cool instruments and techniques, but mass spec just kind of captured my heart because I feel it's so versatile and just such, I guess, marketable skill in a way, in a sense that it's like usable in so many different industries. So it just opened up a way for me to like, maybe today I'm interested in like space, but maybe tomorrow I'm interested in pharmaceuticals.
Or space,
I guess,
pharmaceuticals.
There you go.
One day. Also, I just have an image of a little baby Daniela running with test tubes full of dirt, but also that a professor getting cringe because that just scratches the glass. So I'm scared for all of you. Oh my goodness.
Speaking of scratching the glass, that's how we got a lot of silicone in our NMR spectra in techniques class. We thought we had product, and it was a piece of glass we'd scratched off at the bottom of our test tube.
Yeah, our professor was like, you have to scratch so you start crystallization, and we were just like, nothing's crystallizing, so we keep scratching.
See, I think everyone should listen to this in terms of the fact that these two geniuses, as well as me included, we made a lot of mistakes in this class. Mainly with me, I would mess up the rotovap 9 times out of 10 to the point that I probably should never touch another rotovap for the rest of my life. And that was just with me and my lab partner. I'm not going to say his name because there's no need for that. But, oh my goodness, the humanization is wonderful for this. So, with all of our research going on, and everyone's had a week, I'm sure, describe what you have done this week, whatever it may be, in a song. It can be a favorite song. You might have had a good week. So, what song are you bopping to right now that represents your week, or maybe you're having a iffy week? You know, any one of you can start.
I feel like the song I gravitate towards is Weightless by All Time Low. You know how it goes, maybe it's not my weekend, but it's gonna be my year. I think that one speaks to me because, you know, sometimes in lab things don't work, but, you know, I'm determined to make this my year. It's gonna work.
That's my weekend every time I talk with cells, but we don't have to get into that. Haley?
Yeah, I feel like, you know, we're all starting our 5th year in graduate school, so it's kind of a point where, I don't know about you guys, but I definitely feel like pressure to really make things start coming together, whereas before it was kind of like a little bit easier going of like, let's just try new things and have fun with science. And now I'm like, okay, something needs to work now.
Yep.
I'm going to say I definitely in lab have been listening to the playlist on Spotify called Aloe. I absolutely love it. It's like mellow, but just has all the good vibes. And I think in general, I'm trying to think of, that song. I think it's called The Middle where it's like, it just takes some time, little girl, you're in the middle of the ride. I think it's just, Brene Brown has a quote. I've been listening to a lot of Brene Brown on Spotify too. Has a quote where the middle is messy, but that's where the magic happens. And I kind of feel like I'm hopefully in a part of grad school where the magic will start happening, or I'd like it to happen soon. But it just feels kind of messy too at the same time.
Is it magic or is it science? Who's to say?
Isn't it the same thing?
I mean, it depends. If you watch Rick and Morty, you might have a different opinion, but hey, it's usually magical when you have wonderful people like you two in lab.
Aww.
That's where the real magic is. It's not actually in the science. That's the lie they tell you to get you to go to grad school. The magic's in the friendships.
But you still go. I mean, I don't know. If I had you two in my lab, that Disney playlist would be playing all the time, and I would.
That is what I miss the most is us being silly together in lab and being able to play our song, like music together. My lab is very much a no one is allowed to play their music out loud. Everyone has headphones in and I miss having to put up with each other's music and just learning to enjoy it. Cause Ralph was not convinced by our Disney at the beginning.
No.
He'd go off into his little corner and try to hide and put his headphones in, and eventually he gave in and would sing along with us, but he was not convinced at first.
Well, and then whenever an experiment didn't work, we'd play Bad Day.
Yeah.
Which I think is a thing you have to acknowledge in science. We were talking about this in my lab with some of my lab mates recently of when an experiment doesn't work, your day just kind of feels ruined sometimes, and you're like, okay, time to play a sad song and then move on.
It's funny because I have a video of you singing Bad Day about a gel that we absolutely ruined. I think we were trying to do a stain or something, and it looked terrible.
I think I know what you're talking about. I think we called it, the demon gel. Yeah.
Yeah, and
absolutely.
I love that video, and when I'm having a bad day in lab, I'll pull it up, and it'll, it's a video of a bad day, but it actually makes me smile now, so.
You'll have to send that one to me. I don't think I have that.
I don't think I have that, but I fully remember the demon gel because we hung it up on that board of all of our mistakes.
Yes, we printed a picture of
that one, and then when methanol got into the calorimeter and it just wrecked the entire calibration, and I was just like, wow, I suck at this.
I know we're getting a little bit ahead, but I think it's a good point to say that I think that's what kept us together in our undergrad lab. Was we were laughing at our mistakes, and I think that's so important. It's just it's science, you're gonna mess up, so you might as well just laugh at how horrendously things go sometimes.
Absolutely.
Laugh and also celebrate how far it can go in terms of mistakes, because some of the things we pulled off in terms of mistakes were crazy enough, to the point I think our favorite quote is "everything's on fire." Even though we shouldn't say that in a lab, it's just fun to say. Because, I don't know, methanol was probably the least of my, not the least of my worries when it came to my project. But a number of things could go wrong, and I would just be like, how did I pull that off? How did a hair get in there? How am I actually titrating things with the hair inside of the
Oh my gosh, I love that you kept going, my beard hairs keep getting in my science, in my test tube.
That's the thing. I would literally be working with solution and be like, is that my beard hair? Oh, I gotta start over.
It happens to the best of us. It really does.
It really does. Also, it's not... Okay, back to previous comment. Yes, I was a little bit shaky and not trying to participate with Disney, but mind you all, I also was very annoying with my music taste because they know the one song I played the most in lab.
Molasses.
Molasses by Hiatus Coyote. Because it's such a good song. But to the point that, okay, I think most people, when you have a playlist, if you're going to go throughout the day, you don't start over the playlist. For me, for everything I did, I would just start it over from the beginning. I never let it play through. So they would be running a gel, and it would be like 12:30 after lunch. And instead of me continuing on to my playlist, you hear do 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 do, and I was just like, really? You don't want to do anything else?
You know, the funny thing is, like, I swear, like, you just had this one song on repeat. So I absolutely hated it by the end of the summer. And then I, like, randomly remembered it, like, 2 years into grad school. And I was like, you know, that was actually a pretty catchy song. And I listened to it, and I fell in love with it. And I just fell in love with Hiatus Coyote as a whole. I started listening to all their other stuff. So it ended up being a good thing at the end of the day. But when we were in lab, my gosh. But it's okay. We did the same thing. We did the same thing with Disney music, like "Colors of the Wind" from Pocahontas. Yep.
Well, and to be fair, whenever we weren't listening to Disney, we would put on the radio, and that is just as repetitive. So
yeah.
I think I heard "Timber" by Kesha way too much in our first year together. Great song, just annoying. Between that and Reflection and Let's Get Down to Business.
Oh yeah. To defeat the Huns.
It's funny because one of the very first memories I have of you, Ralph, of like when I think of you as like my friend, like I knew you before, but it was in Techniques, and Haley and I were singing that, and you came up to us trying to like say something or ask us a question, and Haley and I were not having it, and we just like in perfect unison in chorus we're singing to you, "We'll Make a Man Out of You."
I remember that. I was scarred for a second.
We've already gotten off track. We knew this was going to happen, but it happened pretty quickly.
And I'm including all of it while we're talking. Oh my gosh. I think, we gotta continue on this because I think it's an important thing to remember what you missed from undergrad, like learning, but also just lab stuff. I will definitely miss just watching Netflix with y'all because for all of you don't know, I don't understand.
We still do that.
We still do that, but like there was some camaraderie there when I don't understand, like you all, I don't watch Say Yes to the Dress. I don't watch Great British Bake Off because I don't understand it. And so these two introduced it to me in a way where they're like, they know everything. They know what each like chef is going to do, or pastry maker. I don't know the word. Baker. Never mind. There we go. Baker. And so I'll be like, oh, that's weird. I'll just watch whatever I'm watching. But then I would get into it and be like, oh, I see what they did there. The raspberry filling was going to make this color, all that other stuff. And then Say Yes to Dress came, and at the time we had someone who was getting married soon, Caitlin Dobbins, who was another lab member. And I know nothing about wedding dresses. Like, why would you choose that dress over another one? And these two, I guess, were very educated on the topic. And I don't know how you all know this.
We just watch too much of the show, honestly. And I still do. It's like one of my top shows to watch when I just need to de-stress. And I think part of it's because you just get these like petty comments from the family that are like, we don't like what you like. It looks horrible on you. And
sounds like
so it's got a certain pettiness and like easiness to it, but it's also like happy at the same time because these people are excited to get married. So like, and then they like cry and they have their bridal moment, which is like easy to make fun of, but also is like happy and
it's a guaranteed happy ending with like some mild conflict, and it's a good way to get your mind off of whatever. It's like mindless TV.
It just sounds like the social version on TV of like a PhD. Like petty comments, really? Your mom is giving you petty comments for what you like, and your PI does the same thing. Either way. Oh
okay, side note, my pet rabbit is digging in a cardboard box home that he has. And I do think the mic is picking it up a little bit, so I do apologize.
It's okay. Everyone, this is Bean is a legend. Is a lovely legend.
Bean is an honorary lab member at this point, so
Yes.
Bean be out here, and it's Bean. This is the thing. Haley will send Snapchat, and by the way, the only we connect through every social media platform, everyone, but Snap is like the favorite one for these two. I don't. But the only reason I go on Snap is to see videos of Bean, and Bean is either like munching on food or just like hopping around in a circle in Haley's apartment, and I'm just like, this is nice. I know this particular pet. Like, I haven't
He's a
met
nugget.
a precious nugget, such a precious nugget. Oh my goodness. Do we miss Karen? We do miss Karen.
oh my god, so much.
Absolutely. Absolutely.
I think we all need Karen back in our she's in our lives, obviously, everyone, but
She's a good Karen. She's a good
Karen. Oh my gosh. Let us not forget the other memory. The quote board. The great quote board. And for everyone out there, when you work with people for a long period of time, you start to learn the different things that they might say, or something outrageous will come out of their mouths. And we decided to put everything we've said on a whiteboard, and we never erased it. I don't think. Did we erase it?
It got erased at the end of every summer because we just ran out of space.
Yeah, but we took pictures for
Yes, we
for
did.
prosperity.
I think. What? Daniela, you okay?
I'm looking it up. I know that I have a picture of it. I think my favorite one was, I can't find it right now on my phone, but this one is like seared in my memory. I was freaking out about doing an oral for our Furman Engaged, which was like a university-wide conference about research and stuff from all different departments, and I was freaking out about it. And Ralph goes, "We'll be there to support you, even if you fart and fall on your face."
Oh my gosh, yes. Also, okay, this just reminded me because it happened in the same lecture hall, but we were in the middle of someone's presentation and Ralph and I were leaving. It was probably on Furman Engaged, and Ralph's backpack string, one of the cords from his backpack got stuck in the wheel of the chair he was sitting in. And he kept trying to yank the cord out, but it wasn't working. And he actually broke the chair. The wheel came off of the chair, and he still couldn't get it off of the cord. So he walked out of there with the wheel swinging from his backpack and knocking into everything else on his way out. So it was the most loud exit I've ever witnessed from a lecture hall.
Hey, the wheels were falling off on that one.
They certainly were.
I think, and it took me a minute because I did not take it off until we got back to lab, because I had to remove the wheel from the string and then somehow sneak back into the lecture hall to put it back on. I had no shame, everyone. No shame.
Nope.
All right. How about we take a break and we'll be right back? Sound good with y'all?
Sounds great.
Yeah.
Hello, everyone. It's Ralph from the South here with your song of the sound for this episode. As I was thinking about the song that I would have for this episode, being that it's all about community with me, Haley, and Daniela, the song that came to mind is one of my favorites. I hear it at every big family reunion or big gathering with friends or any type of reunion that involves, say, school or work. And it's Family Reunion by the OJs. The OJs are a group that was formed in the late 50s, coming out of Canton, Ohio. And they would then go forth and become probably one of the greatest soul groups that would come into existence within the late part of the 20th century. Hits like Money, 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 Money. You say, Money. But in this case, with Family Reunion, which is a part of their titular album, Family Reunion, Eddie Levert sings these words to us it's so nice to see all the folks you love together sitting and talking about all the things that have been going down. When it comes to me and my best friends, especially during this past year, and even this year as we go forth, it was very crucial for us, even when we began in grad school, to keep communication and tell each other what has been going on. I hope that to you do the same with your family, your colleagues, with your grad school friends, and even friends that you've met throughout college and throughout life. With that, sorry to be a little sappy, but let's get back to the show. You want to hear a song, you two? I have a song for us. You ready?
Oh, I'm ready.
Yeah.
Why do I keep my mind on you all the time? And I don't even know you.
I don't know you.
Why do I feel this way? Thinking about you every day. And I don't even know you.
I don't know you.
In your arms. Fill me with all of your charms. And I'll take to the sky on a natural high.
Maybe you just smell too much chloroform.
Jesus! On the day I die, take to the sky on loving you. You know what? Oh, God.
You didn't even get to the next line. I had one ready for you.
You might as well say it now. Maybe I did smoke too much chloroform. Oh, God.
Oh my gosh, you know what this also reminds me of? This is just turned into reminiscing hour. But when I was like delirious out of my mind from a little bit of sleep deprivation about our stupid analytical reports, and we were going to cookout because I just needed, I think we all needed some comfort food at that point. And Ralph played a song that he had mixed. I didn't know he had mixed it. He didn't tell me. But all of a sudden there's a trumpet sound that it sounded like a fart noise to me, and in my delirious state. And I absolutely lost it and could not stop laughing. I was like literally tears streaming down my face. And then Ralph was like, I made this song, and I felt so bad, but it really did sound like a fart noise. And you know, farts are still funny no matter how old you get. So
that's the bar. Whenever Ralph shows us a new song, we're like, well, at least there's no fart noises this time.
You know what? I miss that. I do. My music has changed a lot since then, you all. Oh my! And
your music is really good. I'm not like bagging on your music. Your music is great. It just, again, in my delirious state, my maturity level downgraded by like a decade.
Yeah, we were in a sensitive place. I think this was the point where we almost broke in undergrad. We had to ask for an extension. Gasp. It was inconceivable to us at that point to ask for an extension and know that we are human and we could not live up to an arbitrary deadline.
Yeah, we were literally like tearing up on the way back to our apartment, I think. And I like got Daniela to play volleyball with me, and she hates volleyball. Like, that's how weird of a place we were in at that time. Which, looking back, I'm like, whatever. But I think, you know, when you're in the moment, and I think this is also true for grad school, it's like hard to put things in perspective. Like when you're constantly surrounded by like academics and stuff, it kind of seems like everything, it's all or nothing, very black and white. And like, but when you put it in perspective of life as a whole, it's kind of like, okay, this is a very small portion of everything. Did that get too deep?
No, you didn't get to do. No, because I'm thinking.
Everyone got real quiet, and I was like, "Oh no,
No,
too philosophical, too
it's
existential."
It's perfect because now that I think about it, I mean, I understand how you feel in terms of asking for an extension, especially where we come from. That was crazy. But even now in science, at least when you're applying for conferences, and I don't know if this is the case for you all, you can ask for extensions and they're willing to work with you. And it's weird because I still remember being in class. I also was the kid in class that, like, if I was late to class, I would stand outside the classroom thinking it was high school. It was like, I cannot go in. And it took professors to be like, you can come in, you're just late. I was like, oh, okay. And so everything changes once you switch from undergrad to grad school, where everyone's like, oh, we get it. Something like that. We get it. There's still, like, some of that, like, you should probably be on time. You should probably do better with your organizing of doing work. But some people miss grant deadlines and somehow they're able to submit still.
I think it's just like reorganizing priorities. Like, you're right, Haley, like asking for that extension was, I remember crying on the way home with you.
Yeah.
And now it's just like, you just gotta learn what is worth your mental energy. Like, I just missed the deadline to submit our annual report, but I am cool as a cucumber because I know no one's going to read it for a while, so it doesn't really matter. It's going to get turned in eventually. It is what it is.
Yeah, I found too, like, there's things I don't want to say purposely miss the deadline on, but like, there's things I'll definitely put off if I think something else is more important. Whether it's an experiment or, you know, something in your personal life. Like, I think there's always work to do, and you just really need to be kind of the owner of your own priority list.
I don't like making lists, though. Why do I have to do that?
Well, I mean, I don't think it has to be a formal list, but just like a decision, really.
No, I got you. Being the owner of your priority list. Ooh, I like that. I like that a lot. We also got off topic, which is good because this is going in still. So, for all of you, if you're still listening, in terms of my shenanigans of singing and having these two lovely women essentially get me off the song, because that was a laugh that I needed really badly. The song of the show for today is called Natural High. Natural High is a song, and actually it's an album as well, by the group Bloodstone. We're going to Kansas City, Missouri, where we're going to find a group of people that got together. It's almost like a 13-piece band that are honestly underrated. They actually were performing with Marvin Gaye. They performed with Curtis Mayfield, Elton John, and Impressions. And it was led and arranged, at least for this album, by the leader of the gang, Charles McCormick. And within this album, they sold 1 million copies. It was certified gold. I thought it was really cool. And one of my favorite lyrics, which goes into what we're talking about today, which is our relationship with science. It's a love and hate relationship a lot of the time. When the lyric I just sang, and I'm not singing it again because who knows, my voice, I might be on chloroform, who knows. Why do I keep on you all the time and I don't even know you? Why do I feel this way thinking about you every day and I don't even know you? There have been many a time when me and my friends have discussed not even understanding our projects anymore, and yet we still love science well enough to go back and not maybe we put out fires, maybe we caused some fires, but we're going back into lab to understand things. For me and my relationship with science, it took me a minute to just accept mistakes all the time. And I've never told the story to people. I think it's the funniest story to me because my mistakes are always the most simple thing, but when you think about it, I took things too seriously. I would spray ethanol on my petri dishes that had cells in them because my PI told me to be as sterile as possible. This is the first time I ever ran into the fact that being too sterile could cause everything to go wrong. So that's my big mistake. But once I got over that, you start learning the different new mistakes that you can make in science. And for me, I work with a lot of cell lines. For these two, actually, I'll let them tell which they work with and what they end up finding that's the troubleshooting time of their lives. So y'all go ahead and explain.
I think, okay, I recently learned to do like mammalian cell culture, and the person who was training me, I actually asked them like multiple times. I was like, are you sure we don't have to spray the plates down? Like, are you sure? So that's absolutely a mistake I would have made had I not had someone like looking over my shoulder, I think. So that's I feel like I 100% would have made that mistake. I think two mistakes, honestly, remembering controls. Like, I mean, I run controls. I think everyone runs controls, but remembering and thinking about every possible control you could have done is something I think I'm sometimes forgetful of. And then I think about a control I should have done, and then I have to run it again later on a gel. And like sometimes things just happen. Like I've been trying to, kind of make some negative stain grids for electron microscopy lately, and for whatever reason, the protein is not going onto the grid. This is a pretty like basic technique to do, and I don't really know what's going on yet, so we're working with that. But you look at the grids and there's just literally no protein there, and I'm like, well, I don't know where it went. I've definitely accidentally bypassed a column on an active before. I have been concentrating protein and then dropped the concentrator while open into the sink on like the third or fourth day of a protein purification. At that point, you're like, okay, we're going home. We're starting anew tomorrow. It's going to be okay.
Oh, jeez. Literally, that moment when you don't even know. Like, you don't even know what's wrong. It's just not the thing you want is, like, the bane of all science troubleshooting. Because eventually you pull in postdocs and they're like, I don't know. And then you pull your PI and they're like, I don't know. It's like, well, who knows? Tell me somebody who knows.
Yeah, I think that's one of the most frustrating things about, or one of the things I really had to get used to in grad school is, in undergrad, when you're like doing math problems for a chemistry class and just doing practice stuff like that, or you're doing a lab protocol like with a group of people, like somebody knows the way to do it. And I think getting to grad school, I still sometimes do this, is I almost try to like over prepare for experiments, and I'll ask everyone advice on how to do it and stuff, especially if it's something new. And I like, in the back of my brain, I kind of expect someone to be able to like guarantee me a result if I do it a certain way. And the reality of grad school is you're doing something no one's done before in your research. So there is no guarantee it will work, and there will be troubleshooting. And I think that can be a really different mindset to get used to and to come to terms with after everything else in our academic career being so different.
Daniela?
Wow. Mistakes. I'm just trying to narrow it down in my head, to be honest. I think one that I am like kind of legitimately embarrassed about in my lab is, so I work with mass spectrometry, as I mentioned earlier, and specifically I do mass spectrometry imaging. And so I work with these really fine, what we call capillaries. It's basically a like hair thin piece of glass that I shoot my solvent through. And in order to connect these really fine pieces of glass to a syringe that has my solvent, we have these little connectors, and they're like made of plastic. And when I first joined the lab, you have pipette tips. Those are disposable because you have like a solvent in them, and then you don't want to contaminate things. And so I had these little pieces of plastic, and I just kept throwing them away after I was done with my experiment every time. you're not supposed to. You're supposed to reuse them. They're like $100 a piece or something ridiculous. So I probably threw out like $1,000 worth of these little pieces before someone was like, what are you doing? Why are you throwing those away?
That's a month's rent.
That happens. I was like, oh, I didn't want to contaminate my site. No, they're like, no, I mean, you're just flushing solvent through them. You just clean them and it's fine. And it's like, oh, whoops. Okay, no one told me that. The most common thing that happens though is, so I'm working with this super fine pieces of glass, and even though they're already really fine, I pulled them into really fine tips. So I can actually create a droplet on a like tissue surface that's about, 15 microns big. It's so pretty tiny. And so you have to be super careful because I'm trying to move this stage underneath these really sharp, fragile pieces of glass underneath. And I have definitely on multiple occasions just clicked the wrong button, smashed the stage up on these really fine glass capillaries, and completely destroyed hours of work. Like, it happens all of the time. This experiment takes like 3 hours to set up. Everything is working perfect. I click the wrong button, smash everything to bits, have to start all over again. So that happens. And I mean, it happens less now with more experience. But when I was first starting, I mean, I would have 3 days on the instrument, 14-hour days, and I would get nothing because I just smashed my experiment to bits again and again and again.
That's absolutely like what I was saying with like, there's on the ACTA, which is like an FPLC system we use to purify our proteins. If you forget to click a button, like the default settings are, as we have set, are to bypass the column. So if you bypass the column and you loaded all of your protein and it goes to the waste, if you didn't put a new waste beaker, you've just basically probably lost all your protein. It can, depending on what's in the waste beaker, it can be unsalvageable.
Well, sometimes it's not even your fault, though. Like, sometimes things go wrong that are not even your fault. So, earlier I said, maybe it's not my weekend, but it's going to be my year. So I'm going through one of the dips of science right now, like not great times. I don't think I've talked to you about this yet, Ralph. I think I already told Haley. I just found out that 3 months of data are absolutely useless, all because the software that I was using is stupid, essentially. You have to click the buttons in a certain order, otherwise it doesn't recalculate the sequence of like acquisition, how it's getting the data. And so even though you think it's doing one thing, it's actually doing another. And so 3 months of data were acquired the wrong way. And in order to be able to use that data, it has to go through another program. And because of that mistake, I can't use any of that data because the program can't visualize it the way it was acquired. And so, 3 months of data, bye. And that just, it happens. Love-hate relationship, but I'm also running, I'm already running new experiments to account for those 3 months of lost data. Because you're right, Ralph, love-hate, you gotta keep going.
It's a love-hate. It's funny because that reminds me, I think I told maybe both of you this or not, but I had my thesis committee meeting and I ultimately learned the same thing, that I've been doing something, or I've been doing the thing at my potential, how I'm supposed to do it. And reviewers, as we all know when we write a paper, they're brutal. They want what they want. They want something night and day if there's a change. And essentially, there's one way of finding out that everything you did was wrong, but there's another thing when they say it's not good enough for us. And it's like, well, what do you want me to do? And it's like, that's up to you. And so now so either you did it wrong or you need to optimize to the fullest way possible. Otherwise, you just have to switch. And it's just like, well, darn. Guess no.
Reviewer 2, man.
Yeah, reviewer 2. Can we also just talk about, Ralph, you can relate to this one. Like, God bless. Like, you're doing an experiment and you know it's right, but your Western blot is just dirty or has a higher background, and you're like, okay, the data is there, but it doesn't look presentable just because the Western blot's messy, and you just have to do it all over again just because, like, something that's not necessarily related to, you know,
you're trying to get something picture perfect. Like,
yeah, exactly.
And I think everyone, I mean, we're learning this as we go through this sheet, like the past 2 years in terms of color, but like, nothing is picture perfect, but the figures we put in paper has to be, if you want something like high impact. And it's scary because, like, our bodies aren't picture perfect. They're always going to change. My cells that I work with, they're never going to be picture perfect. But we have to get it to a point where it's, like, presentable.
Yeah, it's like, you know, everything's got variability in it, and it's trying to figure out what's causing variability and decrease that as best you can. But at the end of the day, especially, I mean, even Daniela, who's more on the chemistry side of things still, and me and Ralph is, I mean, you're dealing with biological samples, and to some degree there's just variability between samples that can be difficult to account for.
Oh, absolutely. And just like, you know, the whole Heisenberg principle, not to get too fancy, but like, are you changing things when you're observing them? Like, that's an argument that I always bring back in lab. And it's like, is this actually what we're seeing, or are we creating this by our technique? And
yes, my gosh, yes.
Yeah. So, science. I think it's been hard to like also accept that things aren't perfect, because I feel like in your mind when you're growing up, you think of a scientist, you think of science, you think of a yes, no answer. Like, it's a binary, and it's really not. It's a spectrum. And I think you mentioned like Corona. I think one of the frustrating things to me at the beginning was when people were saying, oh, scientists don't even know what they're talking about. They keep changing their minds. And it's like, well, if you're a scientist, you've learned that like that's just it. Like, science is like an ever formulating hypothesis. Like, you're always changing your mind. You're always getting more evidence. And it's never perfect, but it's just more and more and more evidence, more of a case to what your conclusion is. And yeah, sometimes experiments don't look great, but they're telling you what, you know, they don't look pretty, but they are adding to that case of evidence. That's why the supporting section is bae. Because that's where I throw all my experiments that aren't pretty, but like, I'm just like, I did a lot of work for this and it supports my story. So I'm going to include this. Supporting section. Be there for me. Support my endeavors.
Okay, I will say I also have a love-hate relationship with supporting information sections, because I think a lot of critical controls and things for experiments end up in the supporting information, because it's what proves that your experiment is valid, but it's not what proves or disproves your hypothesis at the end of the day. And with journals limiting figures and text, which makes sense from a historical perspective when these were mostly printed, but
doesn't anymore.
It isn't really printed. And so I think, you know, the way science is going is just papers are getting bigger and bigger. They involve more collaborations. Author lists are getting longer and longer. The amount of data we have to have in a single paper to get it published is growing. And I think a lot of that gets shoved to the supporting information. And if you want to look through a paper thoroughly, you will go through the supporting information. But on another level, with how the expectation that we have of ourselves, our PIs, everyone, to keep up with the literature, I think it's getting more and more difficult, and you're not going to be as likely to pick up the supporting information and look at that. And I'm curious what that'll do to our, you know, I think it's interesting that it's changing. It's definitely changing how we present our science and publish it.
I think you're right that it's changing. Like, the lengths of paper are getting bigger, more information, like Ralph was saying, like reviewers expect all of this extra info, or like pretty much redoing your entire experiment sometimes with like a different whatever, a control, a different line of cells, something different. But I feel like the expectations in academia are very much staying the same in output wise. So like I think as grad students, even though getting a paper out might still be 3 times harder than it was 10 years ago, they're still expecting the same amount because, you know, your PI might be like, oh, I had this many papers in grad school or whatever. Or like, I don't know, I feel like older people aren't getting with the times on like output expectations, even though the work is significantly more invested now.
Yeah.
Book. They want a book. They want us to publish a whole book on our research right now. Best-selling author right now. Versus back in 1970s when you could get into Nature off of like one figure.
Well, yeah, like we've talked about this, Haley. Like the structure of a protein got you into Nature before.
Yeah, and now with AlphaFold coming out, for those of you who maybe haven't heard, AlphaFold is a program it's owned by Google, and essentially what it does is you can feed it a protein sequence, amino acid sequence, and I think it uses evolutionary algorithms, but I haven't completely delved into the methods of this paper yet. But basically it can predict the structure of a protein with incredibly high accuracy now, to the point that it's basically just as accurate as experiments. And so it's really going to change how we do structural biology. And, you know, the same parallel can be made with the Genome Project. So people used to be able to get their entire thesis done based off of sequencing a single gene. And that's no longer going to get you a thesis project because we have the entire human genome sequenced. We have a lot of other genomes of other model organisms sequenced as well. And so now, at least through the lens of structural biology, what this means is if you get a structure of a single protein, that's probably not going to be your whole thesis project. You're going to need supporting biochemical information, or really what it's kind of leaning towards is getting structures of multiple proteins bound together, or dynamic data, really.
Why can't we get a machine that just does all my Western blots for me? Like, if they can do all this, I'm gonna need Google to just get the Western blot machine going right now. I don't need to...
Okay, do you have one of those dry transfer machines, though? Because that is honestly one of the best inventions I have ever worked with.
I've seen them, but like, you know, in addition to what we just talked about, some of our PIs are very old school, and by old school, like 2000s on up, that you must transfer using this box. But like, even then, if we have the technology to pull off that, what you just said, we have the technology where I can be like, run a theoretical gel for me, please. Oh. But, okay, we've talked about, okay, with all that we have said, we still end up in the lab every day. Why?
Gotta get this bread, Ralph.
Gotta get this what?
This bread. Gotta feed myself every day. No, I'm kidding.
Oh my God.
I'm kidding, but not.
I'm kidding, but not. I don't know. Haley and I talk about this a lot, and I've talked about this with you. It definitely is a love-hate relationship. Haley and I on the reg are just like, okay, how do we get out of science? But also, if there's like a science outreach opportunity, we both jump on it.
Yeah, it's funny because we both love outreach efforts. I think it's super fun. I think it's really creative. I think it's fun to show new people about science and see them get excited about it. But I know, like, we both have frustrations with science, as I'm sure anyone in the field of science does. I don't think you can... I think there's cons to anything and everything in life. And, you know, science is not an exception to that. And we both talk and we're like, if we're frustrated with science, are we like, is it kind of evil of us to be doing outreach efforts and like trying to recruit new people into the field of science on some level? But, you know, I think as frustrating as it is, I think there's a lot of creativity involved in science. And that's one thing that I have a love-hate relationship with in science. At the level of a grad student, you're doing so much wet lab experiments, and a lot of it just comes down to optimizing what's in your buffer. Is my buffer salty enough? I'm always saltier than my buffer, let's be real. But does my buffer have enough salt in it? And there's things like that I just find a little bit, what do you call it? Is it banal or banal?
Like,
I don't know how to say it.
The drudgery of it. Yeah, the drudgery of it. I think the politics of it, the bureaucracy, and just like the system of science, I think is often what gets us down. But I think at the end of the day, we're all kind of nerds. Like, anytime you find something really cool that's new in science, you get really excited. You know, anytime I read about a new technique, I'm like super excited, like, oh my God, this is so clever. This is so clever. Like, they're so smart and it's so cool. You know, but yeah, there's the system gets you down.
Yeah. And I'll say, like, that's what I love about this podcast at you, Ralph. I'm going to shout out to you because I think you keep science fun and light, and you really bring it back to why we love science. Like, I go back to singing Natropsin Like It's Hot when we were in undergrad. I'll let Ralph tell you a little bit more about that, but it just he kind of reminds me of why I love science.
For all of you that don't know, I don't think I've talked about my research that much, or maybe I have in past episodes. I use this drug called natropsin, which I think it's a drug that's from it's a bacteria. I'm trying to remember. It's just from Streptomyces, one of those species. But it's supposed to inhibit these proteins that bind to your DNA. But at one point, I think me and Daniela and Haley were just in the lab, out of our mind, obviously. And I was just like,
Always.
Drop it like it's hot. It works with natropsin. So our group chat became natropsin like it's hot. And
it was also because you were titrating it into your ITC, right? So like you were like, it was dropping, and the ITC, like calorimetry, it's hot. It all fits so perfectly.
And this, and one, I appreciate your kind words, Haley. I accept them. I appreciate that because I like having fun anyway. Like, I think I've said on this many episodes before, I dance in lab while I'm doing experiments. And while they, unfortunately, they're
can confirm. Absolutely can confirm.
Yes.
I do. And most of the time in undergrad, it was the floss dance. And that was just like me thinking and me just being bored. But even in labs that I'm in now, or the lab that I'm in now, 9 times out of 10, I'm either popping and locking while I'm trying to figure out what I'm supposed to do, or I'm in the cell hood, like jumping up and down in my chair, getting ready to pipette something. So like, you can call me hyperactive if you want. It's mainly to get me entertained to do the things that I want to do. But I think you both made great points in terms of the fact that it's not necessarily science that gets you down, it's the system of science that we're in. But it's the love of the creativity of it, as well as just being nerds. Like, we got into this for a reason, of course, whether it's Daniela running around with test tubes full of dirt, or we find something fascinating when we're doing it in high school that makes us be like, okay, I want to pursue this. And for me, I mean, I don't think I've stated it. I got into it I'm a little bit petty, y'all. My chemistry teacher essentially said I would never have a career in chemistry. I also ended up not blowing up in a way that's like explosive, but I essentially burned her hot plate for an experiment where I
How do you burn a hot plate? I haven't heard this story before. I'm here for it.
Y'all haven't heard this story?
I do not think so.
So I did IB, which is International Baccalaureate back in Georgia, y'all. And what I did, a part of it is you do an internal assessment. And so what you did in chemistry class is you had to come up with a experiment that you want to do that's simple enough to explain a concept to people. I decided to do some biochemical type stuff where vegetables have terpenes, which change when you put a vegetable in solution. But no one really discusses like, what happens when you put it in different types of solutions. So I put broccoli and carrots in water and milk and in Coca-Cola, and I just heated it up at a different temperature and saw the different change of colors. So that's what I taught. Unfortunately, when you boil milk, it will overflow. And when I put the broccoli and carrots in, all of the milk just splattered onto the hot plate, and I left this huge black ring in the hot plate. It changed color.
Ralph, you and milk.
I was just about milk thing. Can we just bring it back to your other milk experiments where you forgot to put the milk back in the fridge and it smelled like barf?
Like 4 gallons of milk, y'all, over a weekend in the hot South Carolina summer. That entire lab was rank.
And it was for outreach. It was for
outreach. It was the worst. No, but you know what you also just reminded me of? That time we almost died in lab. We had a bunch of old acrylamide in the fridge that we needed to get rid of. Acrylamide is a neurotoxin, so it'll go through your blood-brain barrier and kill some brain cells in there. So, but it's not if it's polymerized. And so our PI, Karen, shout out, was like, hey, we need to dispose of this, but to do that, I need you guys to polymerize it, and then we can throw away the gel. And, we're lazy. We're trying to cut corners. We're, not looking into this. This is something that we do every day. Like, how bad can it be? And we did, like, a super high percentage of acrylamide. Like, usually it's very dilute with water. And I
think
did realize...
tried to scale it linearly, too, and it shouldn't have been scaled linearly. I don't know.
I really don't remember, but all I remember is that what we didn't realize when we did it in very dilute and small concentrations is that it's a very exothermic reaction, and 2, it gives off hydrogen gas.
Yeah.
And so we had this beaker full of very, very, very hot, and it just kept getting hotter, and oh my gosh.
It happened very quickly, too.
Yeah, and you could see the gas coming off of it, and I just remember, like, putting, like, what was it? It was like some sort of shield thing around it, or like a box.
It had like a massive, I forget what it was too.
It was something
you could see through it to shield with, yeah.
Yeah, because we were so scared. We couldn't move it into the hood because I was scared that if I picked it up, it was gonna like shatter in my hand.
Yeah.
But yeah, I think that was one of the most terrifying moments where I was like, I did something very, very wrong. I didn't look into the science here.
If y'all haven't learned anything from this episode, we have come a long way.
Be safe in lab, y'all.
Be safe in lab.
Yes.
Consider everything, but also don't be so rigid with yourself. Oh my gosh. I remember
another quote from the board. Another day in lab, another chance to die. Lab is dangerous.
Yes.
Be safe. Wear your PPE.
Oh my gosh. I feel like we can go on, but we gotta get to some type of end. With everything we're talking about, I think we've shown the not just mistakes, but also just like the excitement and danger of science life for us three. What is in the cards for us for our future plans, Daniela and Haley?
Oh, that's a scary question, Ralph. So hopefully,
fifth years, we gotta think. Yeah.
I'm talking to my boss next week. I'm hoping that we both agree that I will be ready to graduate in a year. Yeah.
Woo.
So I don't know what that's gonna be. I'm thinking industry probably, although I would like a really cushy government job, 40 hours a week, beautiful life work balance. But yeah, if any of your listeners have a job for me, analytical chemist in summer 2022, hit me up.
We out here.
We out here looking for jobs soon. So I will say my career search has been kind of focused around the fact that I'm a little bit tired of wet lab experiments. I think, and it's funny because some people that's what they love most about grad school. They're like, oh, I just love doing stuff at the bench. But to me, it's almost like I find the results really exciting. I like thinking of the next question exciting. I like analyzing the data. That's the stuff I find exciting. And if you think about it that way, wet lab is kind of what holds you up from getting to analyze results and what holds you up from getting to choose the next thing to do. So I've thought about anything under the sun, from being a medical science liaison to just being a project manager in industry. But I think I've also thought about working for a company that does outreach or developing educational tools for people to use. But I think at the end of the day, I just want to lean a little bit away from wet lab work. So it's been a fun ride, but I'm ready to fondly wave goodbye, I think.
Oh my goodness. So we have someone that's going to be managing and taking control, and then we have the industry, I guess, genius over to the right over here. My right.
I don't know about all that, but let me run your mass specs.
Let me hold your max spec for a second.
Give me your $1 million+ experiments. I'm ready.
Yes.
Oh, this has been a lovely experience. A lot of gems, a lot of comical relief. Because I think all three of us, we are who we are. We're super. What were you about to say, Daniela?
I said we're the Three Musketeers.
We really are.
So which one is Goofy?
You.
Do we really need to say it? Yeah.
You know what?
What question is that?
You know what? Bump that. This has been Scientifically Sound. I'm Ralph from the South. Oh, I forgot to tell y'all. This is episode 10. We out here. We made it to number 10. I never thought we would see the day. So we're in double digits, so I had to bring double the people to interview on this one. And it would be my lovely best friends.
Heck yeah.
Thank you for having us. This has been so much fun. I love talking to y'all every time.
Thank you for having us, and thank you for getting us through grad school. I don't think we'd make it this far without each other.
Amen. I really don't. Yeah, there's times I've just called you guys having a late night lab, and I'm just like, okay, somebody needs to keep me company. Everyone else has left the lab already. Where are my people at?
Is there any... Oh, I forgot. Y'all have to put out your social media presence. So where can
so you can get me that job, listeners.
Yeah. Where can we follow you at, Daniela and Haley?
You can find me on Twitter at D Mesa Sanchez. That's DMESASANCHEZ.
Awesome.
You can find me on LinkedIn or on Instagram at Haley HALEY Stubbs STUBBS. No space or anything in between that. I also have a Twitter, and I don't really check it, and I need to be better about that because I'm told it's important for your career. Who ever thought Twitter would be a career tool? But here we are in 2021. You could probably find me from Daniela's followers. I'm going to go ahead and say that. Follow her first and then find me in her list of followers. It's something with my name in it. It might have a number at the end, I think.
Ralph will tag us on his Twitter posts when he posts about this episode. So find us there.
I definitely will. I also just find it comical that as of late, people have been shouting out your LinkedIn's. Just like, shout out to my LinkedIn or follow me on my LinkedIn. Because that's
I just updated mine, so you should. It's beautiful. I'm so proud of it.
Well, if at the end of the day, if you learn anything, always remember, update your LinkedIn. Please don't blow up anything in lab. Wear your PPE. And remember, it's better to have lab mates. It's always better to have lab mates. They're your best friends. Love you.
Okay, bye.
Bye, everyone.
That's the end right there. Hello, hello, hello. It's Ralph from the South. Thank you so much for listening into today's episode. We had some laughs, we had some deep thought, some emotions here and there. I'm thankful for it all when it comes to me and my best friends talking about our scientific endeavors and journey. If you enjoyed today's episode and you want to follow us, you can always follow Scientifically Sound on Twitter at the number 4, the sci_sound. You can also follow us on Instagram at Scientifically Sound, all one word. You can also email us at number 4 again, the scientifically sound at gmail.com. Please like, share, subscribe, chime in, engage, because we're always thankful for the engagement. Have a great rest of your day and week. Bye.